Absolutely. So yeah, I'm David. I am the CTO of BioSha, and I'm going to talk to you today, obviously, about the same antimicrobial resistance challenge that you've been hearing about for about an hour and a half now. And I'm going to talk both, about our solution to the problem, and I'm going to kind of go through why we think this is particularly difficult, some of the sub-problems involved, and go through how we did some of that stuff, figured out some of that. Real quick, BioSha is a New York City-based startup. We specialize in very complex diagnostics, so mainly NGS and metagenomic shotgun-based diagnostics. And we have an assay out for urinary tract infection. These are our accuracies for taxonomic identification, not for antimicrobial identification. But we've just submitted to New York State to add antimicrobial identification to this. So any of you that were here earlier for Leonid's talk will note that this would probably have been a pretty good assay for his cat because it would have identified both the organism and the resistance that would have gone with that. So the reason I give you this background is I just want to highlight that we're sort of trying to figure out how to put this in the clinic. So based on that, this talk might just be a little bit different with how we think about things and how we're looking at, what we're doing with antimicrobial resistance here. So this basically is our pipeline for understanding antimicrobial resistance here. We start with a raw sequencing input. This pipeline is primarily designed for bacterial isolates, but it also does work for metagenomes in certain cases under certain inputs. We can take a bunch of different types of sequencing data as input, so paired end, single end, nanopore, whatever. And all of that input goes into an initial pre-processing step where we do sort of standard stuff you'd expect. We remove low quality reads, we remove sort of highly repetitive stuff. And then we also remove anything that maps to the human genome, which we found to be a pretty important step. There is quite a lot of sequence usually that maps to human genome in anything that was processed in the lab. And then after we've got our nice, clean, sort of human-free reads, we run this through a species identification and assembly step. Now, with the CAMDA data, we're generally talking about bacterial isolates. So just identifying the species is usually not so difficult for a bacterial isolate. That's kind of easy. What we do is we actually go quite a bit deeper and try to get a really detailed strain identification for these as well. In the case of antimicrobial resistance, some lineages are very much more closely associated with resistance than others. And we do this by both a hybrid KMER approach, sort of comparing KMER spectra across different organisms, and with a CG-MLST of the species ID as well. Once we have our species ID, we do a genome assembly. These could happen in parallel. The genome assembly is actually totally de novo. We then annotate the genome using just some normal prokaryotic annotation tools. And then once we have our assembled and annotated and species ID genome, we feed it into an antimicrobial resistance gene step. Now, this, in contrast to Jack's presentation earlier, we just look for known AMR genes in this genome. We just look for genes that are known to be AMR. We do a little bit of other things like plasmid detection. And importantly, we also identify some related cofactors, things like efflux pumps, transcription factors that are sort of relevant to antimicrobial resistance. And finally, we flag key mutations, key variants that have been sort of flagged in the literature as that. Once we have that, we feed this into a machine learning model, just a random forest that looks at all these features together, predicts whether a sample is likely to be susceptible, intermediate, resistant. And this came up with, I think it was the best performance on the leaderboard with an 89% on the train and 84% on the test set for CAMDA. So that's our tool, pretty standard bioinformatic technique, in my opinion, probably not, the most innovative, but it works well for where things are right now. And that's what we're aiming to do with getting this stuff into the clinic. So what I really want to talk about more is predicting resistance and why predicting resistance as a phenotype is so hard. And you've heard pieces of this sort of put together in other presentations today. So first problem. The databases involved, the databases of known AMR genes, the databases of known AMR isolates, particularly the publicly available ones, are quite small. There are a few thousand genes, few thousand genomes. That takes a lot of effort to accumulate that data, but compared to the diversity of antimicrobial resistance out there, this really is, kind of a small data set. The second problem, phenotype is complicated. While a lot of resistance can be sort of one gene equals a resistant phenotype, there's a lot more resistance that is complicated, that is multigenic, that is based on the presence of variants. Anybody who's worked with tuberculosis knows how finicky TB AMR genes are, for example. And then the third problem is that evolution doesn't stop. This is an arms race. There's always going to be new novel AMR genes out there, and we're never really going to be able to completely catalog everything that's there. So we have these three problems. How did we solve them? First off, we have this genome catalog that we call GeoSeq that we've built out. It's a platform where we've collected a whole bunch of public microbial genomic data We've analyzed about 300,000 public samples, mainly public metagenomes and metatranscriptomes. And we've identified about 24 million microbes in this, and we use this to get a baseline data. It sounds, frankly, like Leonid has done something very similar to this in cabbage, but probably at a much higher degree of polish. And I want to sort of highlight how this works here. As probably not too surprising, but as you add genomes, as you add more examples to your data set, your resistance accuracy goes up. This is particularly true for sort of the regime where you're only dealing with a few tens of examples. And while the genomes and the organisms in CAMDA were pretty common, these are pretty well-known organisms which have relatively a lot of examples, there's a long tail of organisms that can be resistant to antibiotics that have very few reference genomes that are not commonly seen in the clinic. And we're really trying to address that here with GeoSeq, finding lots and lots of these data. So, second problem, complex phenotype. This is basically the theme of CAMDA this year, which is AI and machine learning. How do you solve a complex nonlinear problem? You basically just throw an AI algorithm at it at this point. And what's important here about what we did is we're not just looking at known AMR genes or annotated mutations, but we're looking at strain identification, and we're looking at mechanistically relevant genes that are not themselves AMR genes. And an example of our performance there, we looked at aminoglycoside-resistant Klebsiella, not one of the CAMDA organisms, just something we're doing for clinical stuff. When we look at AMR genes only, we get about 73% accuracy in our model. When we add in efflux pumps and global regulators to this, our accuracy jumps to about 89%, just by including these genes, which are not normally considered to be AMR genes. And then the third piece of this, which is still something we're working on, is looking at how genes evolve. We have built out all our tools to receive feedback from clinicians. So we look at what patients were treated with, what their outcomes were, and we use this to feedback whether there was likely resistance in a sample or not, and are building this into an ongoing database. Our results get delivered both as bioinformatic data and as PDF reports, and we are working on developing some new features around identifying plasmids and identifying antimicrobial resistance in complex communities, mainly metagenomes, essentially. We are deploying this in partnership with Boryung into a remote diagnostics tool we're building out that's going to be tested on the International Space Station eventually at some point in the future. And we're always open to more collaboration. Now, I am mostly just the mouthpiece here, so I want to thank the BIOSHA engineering team who has done a lot of the work here, particularly Gabor Fiedler, who did a lot of the underlying work for this tool. And since this was at least originally meant to be just a 10-minute talk, we are hosting a webinar on August 21st that's going to go into a little bit more detail about how this tool works and how it does, which we would love to have any of you join. With that, I can take questions if there are any.
Thank you, David.
Hi again. Sorry, when we're receiving your pipeline, you have all that processing and the selection of targets before you go into ML. So I was wondering, what about the data leakage? Like, what do you do to mitigate that? I suppose maybe the pipeline, the slide doesn't translate to what you do in reality, but how do you do that?
Why would pre-processing the data result in leakage? I'm not following.
It's one of the principles that validates machine learning best practices. If you pre-process your claim and your test data together.
No. It's single sample pre-processing. It's pre-processing an individual sample to remove human DNA and low quality DNA. It's not co-processed. Yeah, that would be absolutely data leakage. Although in this case, I'm going to note that data leakage isn't possible because we didn't even have the labels for the test set.
Right, but you select specific genes that you know, as in all your samples will have those genes. They might not be present in a test set if you have only that in a proportion.
You mean using a known reference set of genes?
Yeah.
I mean, sure, but that's virtually how all of these techniques work, is you either find a reference set de novo or you use a known one. I mean, again, we didn't have the labels for the test set, so data leakage isn't really possible here. Yeah.
Pretty simple question, but what exactly reference genomes did you use? Did you build your own? Did you get them from a database?
We definitely pulled them from a bunch of databases. So we looked at Patrick, which is reference genomes, not genes. CARD certainly is part of this. We didn't do a lot of de novo discovery of genes on our own here. We did assemble this from a bunch of different sources. I don't think we're using any crazy novel source that other people haven't talked about.
Thank you.
So your input to your model is just like existence of not like known genes and these pumps.
Pretty much. Yeah, I mean, there's
Like
some
binary,
variants.
like yes or no.
Yeah, more or less. We have some, additional features for like SNPs and indels and whatever. But yeah, it's a pretty simple input. It's not putting the sequence data into the model. It's just like I think it's like about 100 features overall.
Okay.
Yeah.
And these pumps, why are these pumps discriminative? Like they're like pumps specific to like pumping out certain antibiotics?
Yeah, so I mean, efflux pumps are quite a general class of proteins. They just remove stuff from the inside of the cell. There are efflux pumps that sort of on their own cause antibiotic resistance, and those are pretty specific to pumping out antibiotics. The efflux pumps that we're talking about here are more general. They just remove stuff from inside the cell. And yeah, you could see how they would be related to antibiotic resistance. If they're removing the antibiotic from inside the cell, that's clearly going to be relevant. But if you only use those efflux pumps, they're not terribly predictive on their own. So in this case, it's looking at a multigenic factor. I think actually you highlighted that sort of Neisseria and Pseudomonas were tricky points, and I saw that on a few other slides too. Neisseria was actually clearly sort of multigenic for us, and there were, you had to look at a couple different gene classes in concert to see resistance, and they had to sort of both be present.
Oh, really? Okay.
We didn't have an amazing score on Neisseria either, so we don't have it solved. Yeah.
Any more questions? If not, then thank you, David.